But believe it or not, we've covered a lot of the material that in some of those lectures that look like we're behind. So you're not as far behind as you think. What I'm going to be doing today is kind of finishing off some material on control systems. And I'm going from there to discuss the groups of hormones that we've in fact been talking about. I want to do that before I get into the cancer biology, just to keep kind of continuity with what we've been up to. That means that there's going to be like 4 or 5 lectures worth of material. You're going to see collapse into about 2 lectures of material. And you'll see it fits together quite well in that way. So we're not as far behind as you might think in the syllabus. So I want to begin where we picked up, or we left off last time, talking about controls of the gonadotropins. We started with LH. We started with a relatively simple control system in the male, and the fact that that particular simple control system leads to a problem if you try and control this system externally. And that's because if you cut off something in this particular control loop, you have effects peripherally. And there's no real easy way in order to interfere with this loop without interfering with things outside of that loop. So secondary sex characteristics and sexual potency are directly affected if you affect anything in that loop. So how have physicians, scientists been trying to approach male contraception? Well, the usual way is to try and block this loop either by giving an analog of LHRH that is an antagonist to its normal actions. So if you modify this molecule by changing amino acids, for instance, you can get a molecule that will sit on the receptor for LHRH at the level of the gonadotrope, but won't allow production of LHRH. You could do that. You could potentially give an antibody against LH, but that has problems later on. So this is one of the best ways to do it. You block here, but then you give back testosterone externally. Now remember, I told you that testosterone gets concentrated in seminiferous tubules by the production of androgen binding protein from the Sertoli cells. Well, and that provides a high concentration of testosterone to support spermatogenesis. But if you give testosterone in the periphery, it helps shut this part of the loop down. But depending on how much you give, it probably isn't enough to support spermatogenesis centrally or in the testis. So you may be able to support secondary sex characteristics, but still have a holdup in sperm production. As a couple of students after the lecture last time were talking, though, the window or the amount that you have to give to shut things down here and still support secondary sexual characteristics without stimulating spermatogenesis is very narrow, and it varies from individual to individual. So it's a problematic kind of approach to controlling male reproduction. Sperm are produced in the millions per day. Single ejaculate from a single male, maybe 200 to 400 million sperm. That's a lot of sperm. And he's producing them every day of his life during adulthood. Now, let's talk about the female side of things. LH has multiple targets in the female. Okay, this is a diagram showing part of an ovary. That ovary has multiple cell types. There's background cells, stromal cells. Those will produce estradiol in response to LH. There is this temporary organ that's made after ovulation. The corpus luteum isn't permanent. It's not always present. It normally lasts somewhere around 10 to 12 days in primates after ovulation. It's essential, however, to produce the progesterone, this steroid, that's necessary to condition the lining of the uterus to accept a fertilized egg. If this progesterone isn't there, or hasn't been produced, you will not get successful implantation. You will not get a pregnancy. Progesterone is always negatively feeding back on the production of kisspeptin and LHRH, and therefore LH. So that's a clean negative feedback loop. The other targets, so you've got stromal cells, the corpus luteum. The other targets for luteinizing hormone in the ovary actually only occur in a few days surrounding ovulation, just before ovulation. One of them, which is the longer, is a target for a longer period of time, are the thecal cells, which actually exist just on the outside of a layer of modified tissue that actually forms the follicle wall. So these cells are of the extra follicular tissue just surrounding it. There's blood vessels that go through this area. There are no blood vessels in the follicle itself. So these thecal cells are targets for LH, and they again make estradiol. When we get to talking about steroid production, you'll see that the way they make estradiol is not just taking a substrate and making that steroid. They actually have to coordinate their actions with another cell type. The other cells that respond to LH are what are called the mural granulosa cells. The granulosa cells are the supporting cells in the female. They support oocyte production and development, and they make a lot of the material that ends up in the follicular fluid. They also make estradiol in response to LH, but they only do it in a couple of days just before ovulation. I mentioned that these are the mural granulosa cells. There's another set of granulosa cells immediately around the oocyte. Those are called the cumulus granulosa cells. Those are never responsive to LH. So you've got these are LH target just prior to ovulation.
Depending on the timing.
Cumulus granulosa. Not LH target. And the reason they're not an LH target is that their development tends to be suppressed by substances that are coming from the oocyte itself. There's a growth factor called GDF9 that probably plays a role in that. So these act as if they're immature granulosa cells, you'll see later that all of these are responsive to FSH, but these are ones that never become responsive to LH. So, the only thing that you have to bear in mind in the female is that estradiol negative feedback switches just prior, in about the 2 days prior to ovulation in the primate. A switch like that does occur in other species. The timing is a little different. But the whole idea is that you go from suppressing LHRH production to stimulating LHRH production. And that occurs largely at the level of the neuroendocrine cells that produce kisspeptin. So it's actually, the target would preferably be kisspeptin neurons in terms of that switch.
And that's only for primates?
That switch, the switch?
Yeah.
No, the switch is not only for primates. It does occur in other species.
But the 2 days, the timing.
The 2 days, the timing, the 2 days is largely for primates. Okay.
And it acts on kisspeptin in multiple species?
Yeah, the reason it's on this diagram is largely because it's found in virtually all species that have been looked at. So the axis in the female is much more complex with respect to LH than it is in the male. That plays a role in potentially manipulating the cycle. Recognize that you can manipulate this cycle as you did with the male at the level of LHRH. You can also manipulate it with any of these steroids, recognizing that these steroids not only act centrally, but they also act in the periphery, as testosterone does in the male. So, if a woman is responding to these by changing her behavior, by changing her breast tenderness, for instance, that will show up. So you potentially could manipulate those steroids to control that cycle. I'm going to go to the next one. This is the control of FSH. Now, follicle-stimulating hormone, remember, is produced by gonadotropes, same cells that makes LH. And indeed, people have looked, and it is indeed the same cells. It's not just the same cell population. Recognize there's a difference between those two. In one case, it could be same population, but these cells only make FSH, these cells only make LH. Indeed, it's not that. The cells make both hormones and can make both hormones.
That's in the
Yeah.
males?
Pardon?
You're talking about in the males?
I'm talking about males and females. Okay. So, whoops, where are we? Okay. So the kisspeptin, LH, should look familiar, same controllers, gonadotropes, but this time producing follicle-stimulating hormone. In the male, again, the male side is a bit simpler than it is on the female side. In the male, FSH has one target and one target only, and those are Sertoli cells. Those Sertoli cells are supporting sperm production. And as they're doing it, one of the products that FSH stimulates production of is androgen binding protein. So here, LH and FSH are acting synergistically with FSH making the androgen binding protein, the Leydig cells responding to LH to make testosterone, which can bind here and help support spermatogenesis. Spermatogenesis does not work without enough testosterone. You remove the testosterone, you'll shut it off. Virtually all species looked at. FSH is most operational, particularly in the primate, in the developing primate. This stimulus is less important in the adult. And the reason we know that is because early experiments showed that if you took out the pituitary, so you're taking out the source of LH and FSH, and you gave back LH, it supported testosterone production. But it didn't support, it did not support spermatogenesis adequately. You get about 80% suppression if you knocked out LH or testosterone, pardon me. If you knock out, oh, okay, hang on, back off. If you were to if you give, pardon me, this is the way it goes. If you give testosterone exogenously to an intact animal, you can suppress FSH production, but only 80% of it. So if you're measuring FSH in blood, you give testosterone, you can suppress about 80% of it. So there was another controller that we were missing, and it turned out to be a protein hormone called inhibin. Now, inhibin comes in a variety of flavors. Alpha inhibin and beta inhibin, or A inhibin and B inhibin. The male is making B inhibin. This protein in the male targets the gonadotrope and selectively suppresses FSH. It does not affect LH. And that inhibin is produced by Sertoli cells in response to FSH. You can do that in culture, you can do that in whole animals. So if you combine testosterone and inhibin in the appropriate amounts, you can fully suppress FSH in the intact animal. So that's the male side. On the female side, FSH has one cell type that it targets. Remember, on the male, one cell type, Sertoli cells. On the female, one cell type, and that's the granulosa cells. It's both the mural and the cumulus granulosa cells. And FSH binds to those granulosa cells whether they're in early stage follicles or late stage follicles. So one cell type. The cell types here produce alpha inhibin. So males B, females A. That inhibin also suppresses FSH at the level of the gonadotrope, not centrally, level of the gonadotrope. FSH helps stimulate the production of estradiol, and it does so by having granulosa cells cooperate with thecal cells so that estradiol is produced. And again, you've got the mid-cycle switch from negative feedback to net positive feedback in the female. So combination of LH and FSH in the female to produce estradiol and LH stimulating corpora lutea to produce progesterone. So when it comes to in the female, in particular in the female primate, when it comes to ovulation, you're ovulating 1 to 2 eggs a month. That's as opposed to a rat that may shed 20 eggs a month. Not a month, 20 eggs every 5 days, I should say. Okay. So, but the numbers are low relative to sperm production, which is those reasons, the fact that you've got multiple steroids involved, and the fact that you've got a low number of cells to block, is the reason that the early investigators targeted chemical contraception to the female rather than the male. So if you manipulate the steroid levels, you can manipulate the LH and FSH production and therefore suppress ovulation in the female, which is what's done chemically. Is that the optimum? Probably not. But that's where it stands currently. Let's see what we've got. I'm going to skip this for right now. Okay, prolactin is the last loop you need to look at. Prolactin is made by the lactotrope, also known as the mammatrope. It's a single chain protein hormone. You'll find out it's related to growth hormone. It's having pleiotropic effects in the periphery. It helps support white cell production and actions. It's stimulatory to the secretory epithelium that makes milk during lactation. It's stimulatory in many species, though not all, to the production of progesterone by the corpus luteum of the ovary. There are multiple potential controls of the lactotrope, the most important of which, however, is dopamine. And notice it's a negative, it's a suppressor. Somatostatin, that small 14 amino acid long peptide, suppresses here as well as it does in growth hormone, but this is a secondary control. Another secondary control is thyroid releasing hormone. Part of those slides that you saw just before that I skipped over talked about some work we did on a group in New Guinea that in fact had marginal thyroid function, and so the question, and they also had constitutively elevated prolactin, and the question was there a link between the two? And the main link we were looking at the possibility of is this control by TRH. In short, the result from that particular set of experiments was that TRH was not, in fact, the reason that the prolactin levels tended to be high. Oxytocin, which is also secreted during lactation and late in pregnancy, is also stimulatory toward the lactotrope production, as is estradiol. So during periods of high estradiol, you're going to see lactotrope stimulation and production of prolactin. Stress helps regulate prolactin levels by suppressing dopamine. This is an interesting axis for a variety of reasons. I've shown controls here largely for the mammal. In other species like fish and birds and reptiles, prolactin is often involved with what's called broody behavior or reproductive behaviors. So there's something called water drive in amphibians and reptiles. For species that would lay their eggs in water, that seems to be associated with prolactin levels. It also tends, in birds, will help in the production of what's called a broody patch on the breast of the animal, causing sloughing of feathers, and in some species there's something called a crop sac, and it stimulates epithelial cell differentiation in the production of something called crop sac milk, which can substitute for other foods for young birds. So it's an interesting hormone in that regard, in the wide spectrum of its effects. Okay, the other thing I wanted to point out, dopamine turns out to be one of the hormones that's most affected in conditions of chronic stress associated with migraine headache. Anybody here ever had a migraine headache? Pleasant experience?
No.
Very not very. Sometimes you frequently will have heard of them as sick headaches, headaches where people avoid light, sound, and a lot of it has to do with controls on the blood flow to the brain. And the drugs that are often given are dopamine antagonists. Or controllers of dopamine production. There is another situation that arises and is related, and that is a condition called ergotism, where ergot alkaloids will in fact act as not as dopamine suppressors, but as dopamine agonists. So stimulate functions as dopamine would. In the cases of ergot poisoning, you can get a condition that will suppress prolactin production. Yep.
Prolactin influences like the secretion of the milk from the breasts, or is it
It's
just...
actually stimulating these cells to produce milk.
Okay.
And we'll actually talk about that later.
Okay.
Oxytocin stimulates the cells surrounding these cells. These cells, secretory epithelium, are arranged in what are called alveoli, or little sacs. And the myoepithelial cells are arranged around those sacs. So prolactin stimulates milk production and secretion into the alveoli and the ducts that lead from them. And the myoepithelial cells contract in response to oxytocin and cause milk ejection.
So in females who have just like had a baby
Right.
who can't produce enough milk, is that a low prolactin levels, or is it like totally something?
It may be low prolactin levels, it may be high progesterone levels, you'll find out. It may be associated with oxytocin problems. So there are a number of things that can come into play. So this was the last loop I wanted to show you. Let's see. Let's skip that and get back to other slides. All right. Now, I told you we're going to talk about hormones as groups. The first group we're going to talk about is LH, FSH, and TSH. So the trophic hormones. In addition, there's one other member of this group called HCG, or chorionic gonadotropin. Those all are related to one another. And the way they're related, turn on the light. On the light. The way is they all have an alpha subunit and a beta subunit. The alpha subunit is the same for all the hormones. Within a species. All right?
All of those hormones?
All those hormones share the same alpha subunit within a species. So, if this is a rhesus monkey, now rhesus monkey has the same alpha subunit in all those hormones. If it's a sheep, it's the same alpha subunit in all these hormones. Sheep don't have chorionic gonadotropin. Okay. So, within a species, among species, there's some variance in the alpha subunit. So the sheep doesn't have the same exact alpha subunit as the rhesus monkey or the human or the blue whale. All of them have slightly different alpha subunits. It turns out that those subunits are sufficiently similar so that if you swap in a non-species specific subunit and with a purified beta subunit and allow those two to form a non-covalent, they're non-covalently linked by the way, form a non-covalent intact hormone, it will still function as if it were an intact hormone. It may have a slightly lower activity than that purified from a particular species where the beta subunit came from, but it still will work. So ovine alpha subunit with human beta subunit will still give you a human-like activity for whichever of these hormones you've chosen. The beta subunits are specific for the hormone and determine hormone type. So the beta subunit from LH, I can combine it with the alpha subunit from any of those, and I'll still get LH-like activity. If I do that with the beta subunit from TSH, I can get TSH-like activity regardless of where I got the alpha subunit. Again, species to species, there's going to be some variability in the beta subunits. But the overall effect is going to be similar enough so that if I inject, say, ovine FSH, that is from a sheep, and I inject it into a pig, that pig will act as if it got an injection of its own FSH. If you did that repeatedly over time, you're going to get antibodies against the non-idiotypic, the non-self subunit. Now, so what's this diagram about? The alpha subunit is unique for a species, but common among the glycoprotein hormones. That's the other thing these are. These are glycoprotein the subunits. If you stretched out the amino acid sequence, there are modifications on a number of the amino acids to put on carbohydrate side chains. And they vary among the particular hormones you're looking at. The beta subunit of chorionic gonadotropin is the most highly decorated of these beta subunits. The beta subunit is unique for a given hormone and determines function. They're non-covalently associated, and I talked a little bit about various species. So you've got human, ovine, bovine. This is normally caprine. What's caprine? Nobody know what caprine means? Anybody do their horoscopes?
Oh, like a Capricorn?
Capricorn, okay. What is it?
I forget.
Goat. Okay. Goat. Some people use this for chicken as well, so this can be confusing. M can be murine, referring to mouse, but it also can be macaque. P is porcine. You'll see a variety of those things. If you were to go out and try and purchase these for self-injection, stay with human. Okay. The other thing I need to mention about in this group is that LH and HCG, chorionic gonadotropin. Chorionic gonadotropin is made by the late trophoblast, which is the stage of the developing oocyte, developing embryo that enters the uterus and implants. So this is the late stage of the implanting oocyte. It's also made by the placenta, which is where most of it comes from during pregnancy. So, this particular hormone, again, it's the alpha-beta subunit. They have the same alpha subunit. I'm going to do this right. HCG, however, has a longer beta subunit. It's got about 30 amino acids extra, and it has more carbohydrate side chain. The circulating half-life for this hormone is about 24 hours. The circulating half-life for this hormone, so if you were to inject it and then take blood samples looking for how much is still in circulation, this is about 20 minutes. This one, 24 hours. And it's due to this extra carbohydrate on that beta sub chain. It has other implications for how you detect these hormones in circulation as well. Some of you folks in lab have learned about that.
Isn't prolactin a trophic hormone, or is it not in that group?
Prolactin isn't in this group for reasons you will see in a few minutes. Okay. Okay. So,
Did you say that the beta subunit has a half-life is less than the other one, than the other subunit?
no. This hormone has a half-life of about 20 minutes in circulation. This one is about 24 hours. Yeah.
But this is the beta carbon.
In the previous
The carbohydrate.
illustration, you had that it was early trophoblast, but now you're saying it's late. I just want to clarify to see which one it is.
Where did I say early trophoblast?
In your previous picture.
Just a second. Where am I? No, that's not it. 69, okay.
Yeah, it says early. You
Up
scroll up.
top.
Oh,
Placenta and early trouble.
early trophoblast. Definitely in the late trophoblast, the earliest you'll pick this up, the earliest been seen. If you take an embryo in culture, you can see production of chorionic gonadotropin between days 5 and 7. If you try and sample blood in the mother, the absolute earliest you'll ever see it is day 7, and it's more likely day 10. Okay,
That's what they use for
after
pregnancy
ovulation.
tests, right?
Pardon?
Is that what they use for?
Yes.
pregnancy tests?
This is what is normally monitored for pregnancy tests. And it's monitored, and the only reason it isn't even better than it is, it has to do with the establishment of blood flow connections with the embryo, because the embryo develops in an avascular space until it attaches to the endometrial wall. And it's only after that's established and blood flow occurs that you get a good chemical communication between maternal blood and what's going on with the embryo. So, in early trophoblast, yeah, you could potentially see it, but certainly during the trophoblastic period. That's the key. Thank you for keeping me honest. Okay. Oh yeah, this is the one I wanted to make sure you knew. LH and HCG bind to the same receptor. Okay. And it turns out that an early signal to the establishment of a pregnancy is HCG starting to take LH's place in control of progesterone production by the corpus luteum. That switch is sometimes called part of the maternal recognition of pregnancy. If you try and purify these, by the way, if you try and purify hormones from biological materials, you normally start out with a real mishmash of stuff that is relatively low in effectiveness. The amount of biologically active hormone on a milligram of protein basis is quite low. So if you put that into a bioassay, you'll get an assay, but the curve will be shifted way to the left. Let's see. Pardon me, to the right. Show you what I mean. This is assay response. This is maximal. This is 0. This is amount of hormone prep used. If you've got a very impure hormone from a hormonal prep, the curve will go up over here, and it may only go up part way. So this is a crude hormone from a biological mixture. If you purify this by running it over purification columns, affinity columns, you do something that purifies it, it'll take less of that preparation to stimulate the same amount of biological response. And so the curve may come up like this. And finally, if you have an absolutely pure hormone, the curve may be a nice, beautiful prep like that. Recognize that historically, when people only had biological sources, they were forced to undertake purification as the only way to get a pure preparation. And in order to try and standardize the way people did things, at some point, somebody decided, well, this looks pretty good. And they referred to this as a standard preparation. And there were international standards for LH and prolactin, growth hormone, thyroid stimulating hormone. And that standard preparation was defined as concentration of hormone, of X hormone, and these were almost all proteins, some peptides, but never steroids and things like that. Preparation of X hormone to give one defined unit of activity. So they got together at a meeting and decided what one unit of activity was and what preparation was the best one to use as a standard against which to calibrate everything. And so this is where you came up with international units. So international units. Most of the hormones nowadays in pure form are going to be given as international units per milligram. You will see that unitage on insulin injections. You'll see unitage similar to that for LH, for HCG. They all refer to this purification process and this standardization during the history and development of information on these hormones. Recognize that an international unit varies across hormones. So international unit of insulin is not the same as the international unit for parathyroid hormone. Nowadays, because we've cloned the genes, we can get pure material. That pure material is good as long as it's in at that experimental tube before we start an animal experiment. Once it's injected, this starts to undergo modification by the animal. So you get a mixture of intact, biologically active hormone and biologically inactive hormone. But that's the origin of international units. Where are we time-wise? Okay. Next time what I want to do is to go over prolactin and growth hormone